Hello students, welcome to EPG Pathshala. I am Dr. Chirantan Rawal from APJ Abdul Kalam Government College, Silvassa, UT of Dadra and Nagar Haveli. Today we are going to discuss about a module that is regulation of glycolysis, which is under paper carbohydrate metabolism. So students, let us see what are we going to learn in this module. Our objectives are to understand importance of glucose and its metabolism, to study the glycolysis pathway, to study glycolytic enzyme and the checkpoints, to learn about energy account of glycolytic reactions. We will see the breakdown of glucose and its energy synthesis. Today, I will brief you about glycolysis and its regulation mechanism. In today's lecture, we will see importance of glucose in our body, the process of breakdown of glucose molecule, that is glycolysis. Glycolysis is one among the most important source of ATP. Input and output molecules of glycolysis are also important since it is a major source of energy. Parallelly, it will provide various metabolites that carry out various other metabolic processes. It reserves almost priority in a cell as it gives ATP, pyruvate, NADH as output molecules that on a later stage again contribute in energy generation. My dear friends, this NADH, ATP, pyruvates are very important for the metabolism. Number of enzymes act on a glucose molecule to convert it into a smaller substrates, which in turn can further be hydrolyzed by enzymatic reactions. Glycolysis has a total of 10 reactions that satisfy all the energy related needs of a cell. Broadly, glycolysis is divided into two main phases. Energy consumption phase, where energy is consumed, that occurs during the first 5 reactions, while another phase, that is energy synthesis phase, where energy is synthesized in the form of ATP that comes into the next 5 reactions, that is from 6 to 10. We shall see each of these key enzymes, their reactions, its mechanism of actions and regulations. Last but not the least, we shall see energy account of overall all pathways. This is a concept map of glycolysis. We will first study briefly about the glycolysis pathway. We will then study the various checkpoints, precursors, molecules and the fate of various intermediates. Last but not the least, we shall see the actual biochemistry behind this reaction, the role of various biocatalysts in the glycolytic pathways. As you can see in this concept map, there are various reactions which are irreversible and can be used as a regulatory point to control the overall metabolism. Moreover, glycolysis also helps in release of ATP from carbon, thereby helping with body energy requirement. Since no oxygen is required in glycolytic pathway and glycolysis being common to both aerobic as well as anaerobic respiration, popularly it is called anaerobic phase of aerobic respiration. The word is derived from the Greek term sweet. The glyco means sweet, while the lysis means solubilization. Glycolysis essentially means glucose plus lysis, that is a breakdown of glucose. This is one of the most ancient pathway known to humankind. There are series of biochemical reactions through which one molecule of glucose is metabolized to yield 2 moles of pyruvate and 2 ATP as net energy yield. In this diagram, you get the idea that net reaction yields a 2 ATP molecule when one molecule of glucose is metabolized by the process of glycolysis to 2 pyruvate molecule. Over here, glucose is 6 carbon containing compound, while the Pyruvate is 3 carbon content. Preliminarily, glycolysis is a process that takes place in the cytosol. Both aerobic and anaerobic organisms have this pathway for glucose metabolism. This process is regarded as anaerobic as oxygen is not required at any stage of glucose metabolism. However, even in aerobic organism, glycolysis function in the same manner as presence of oxygen does not affect the reaction. So we can say it's neither aerobic nor anaerobic. This part indicates the fate of pyruvate. The pyruvate can undergo aerobic metabolism, it may undergo anaerobic metabolism. The anaerobic reactions are popularly called fermentation, that is pyruvate to lactate, that is lactic acid production, or pyruvate to ethanol, that is alcohol production. Majority of organic acid are produced from sugar fermentation in this manner, while a fat of pyruvate under aerobic condition generates ample amount of ATP as a pyruvate is completely oxidized to carbon dioxide, which generates higher amount of ATP. In the late 18th century, Louis Pasteur carried out extensive research on yeast and studies its fermentative reaction. Later, in 1897, Buchner moved one step ahead and brought about fermentation using cell-free extract and launched the branch of biochemistry, wherein various metabolic processes can be studied under the in vitro condition. In the latter half of 19th century, a complete map of glycolysis was elucidated. There is a contribution from Amden, Newberg, and Trodiodrov. Glycolysis is a pathway has a total of 10 reactions, at the end of which 2 moles of pyruvate is formed from the single glucose molecule. Below mentioned are the reactions and enzymes involved in it. Glucose to glucose 6-phosphate is a phosphorylation reaction requires energy from ATP. The reaction is irreversible and is catalyzed by enzyme hexokinase. Glucose 6-phosphate to fructose 6-phosphate is an isomerization reaction which is again reversible and catalyzed by glucose 6-phosphate isomerase enzyme, which is also called phosphoglucomutase. Fructose 6-phosphate to fructose 1,6-biphosphate is another irreversible reaction of this pathway and is catalyzed by phosphofructokinase enzyme utilizing energy from ATP. PFK is an important in the regulation of glycolysis. Fructose 1,6-biphosphate to 2 molecule of glyceraldehyde 3-phosphate. Fructose 1,6-biphosphate actually splits into the 2 molecule of 3 carbon moiety, that is an aldehyde and a ketone, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by fructose bisphosphate aldolase enzyme. This explains 2 molecules of glyceraldehyde 3-phosphate to 1 molecule of 1,3-biphosphoglycerate. This 1,3-biphosphoglycerate converted into phosphoglycerate by an exergonic reaction which is catalyzed by phosphoglycerate. 3-phosphoglycerate to 2-phosphoglycerate reaction where the phosphate group shifts from the 3rd position to the 2nd position. The reaction is isomerization reaction which is catalyzed by phosphoglycerate mutase. This 2-phosphoglycerate is converted into phosphoenol pyruvate by an enolase. This phosphoenol pyruvate is converted into pyruvate by an irreversible reaction which is catalyzed by transferase activity followed by dephosphorylation reaction. So now students, let's see the fate of pyruvate. Now what happens with the pyruvate at the end of the reactions? All the enzyme catalyzed reactions can be controlled in some or the other way. Thus we say that there are certain checkpoints that control the reaction. We will here study various such checkpoints. These checkpoints are very important, my friends. These checkpoints in form of precursor molecules and fate of various intermediates. As the concept map in following slides suggests, there are various reactions which are irreversible and can be used as a regulatory point to control overall metabolism. These checkpoints are the regulatory points where we can control the metabolism. Moreover, glycolysis also helps releases ATP from carbon skeleton, thereby helping with body energy requirement. Since no oxygen is required in glycolytic pathway, and glycolysis being common to both aerobic as well as anaerobic respiration, popularly it is called anaerobic phase of aerobic respiration. So in a nutshell, glycolysis is neither aerobic nor anaerobic. Preliminarily, glycolysis is a process that takes place in the cytoplasm. The location of the glycolysis is cytoplasm. Both aerobic and anaerobic organisms have this pathway for glucose metabolism. This process is regarded as anaerobic as oxygen is not required at any stage of glucose metabolism. However, even in aerobic organism, glycolysis function in the same manner as the presence of oxygen does not affect the reaction. That's why I told glycolysis is neither aerobic nor anaerobic. The anaerobic reactions are popularly called fermentation. Like pyruvate is being converted to lactate, that is lactic acid fermentation, or pyruvate being converted into ethanol, that is alcohol production. Yeast is doing the anaerobic respiration where the pyruvate is being converted into the ethanol by the process called fermentation. Similarly, in our muscles when we run, the pyruvate is being converted into lactate. Majority of organic acids are produced from sugar fermentation in the same manner. While fat of pyruvate under aerobic condition generates ample amount of ATP, as pyruvate is completely oxidized to carbon dioxide and water, which generates high amount of ATP. So now students, we will look for the checkpoints of the glycolysis. There are total 10 reactions in the glycolysis, and some of these reactions are irreversible, which act as a checkpoint for the regulation of overall process. In glycolysis, the reaction is catalyzed by hexokinase, that is the first reaction. Phosphofructokinase, pyruvate kinase are such irreversible reaction and can behave as a checkpoint for the regulation of the entire pathway. So these three enzymes carry out the key irreversible reactions which can be checked, which can be controlled. These enzymes are regulated by various factors such as availability of the substrate, concentration of the enzymes, allosteric regulation of the enzymes, covalent modification of the enzymes, and the transcription control. All of this helps in either regulate the glycolytic pathway within the milliseconds to minutes to hours depending on the concentration of glucose within the cell. Glycolysis involves many enzymes and there are certain checkpoints where we can regulate the glycolysis. We just briefly review the glycolytic pathway. We observed that there are certain irreversible enzyme catalyzed reactions which have a negative delta G. This can be potentially regulatory site of the pathway. The preliminary function of glycolysis is to produce energy. It must be regulated so that the energy released in the form of ATP under needful situations only. In glycolysis, the reaction catalyzed by hexokinase, phosphofructokinase and pyruvate kinase are such irreversible reactions and can behave as a checkpoint for regulation of the entire pathway. These enzymes regulated by availability of the substrate, concentration of enzymes responsible for rate limiting steps, allosteric regulation of enzyme regulates pathway within the milliseconds, covalent modification of enzyme, that is by phosphorylation, which regulates the pathway within the few seconds, and transcriptional controls, which brings the regulation within hours. So it depends on the kind of requirement, where if the control is required immediately, then accordingly the system selects the different kind of regulation. The intracellular concentration as well as transcription of all three enzymes is well regulated by hormonal control. Hormone pair of insulin glucagon secreted by pancreas in response to sudden rise and fall in blood glucose level. Insulin is also released in response to sudden rise in amino acid levels in the blood. As a universal effect, insulin promotes the storage of excessive energy under the fed state, while the glucagon acts as antagonist to insulin in every manner. Insulin promotes the transcription of a glucokinase, that is hexokinase, phosphofructokinase and pyruvate kinase, while glucagon demotes the transcription of these three enzymes. So, quite clear that both have opposite actions. The visible effects can be seen when an individual is well fed or is continuously starved. So, there are checkpoints. We can say it's the regulatory checkpoints, and these checkpoints are the irreversible reactions. Checkpoint 1, that is a hexokinase, which can be regulated. Checkpoint 2, that is phosphofructokinase, which is also regulated. And checkpoint 3, pyruvate kinase. These all enzymes catalyze the irreversible reaction. So, their regulation affects the overall glycolysis process. Let's first try to understand the regulation of hexokinase. Hexokinase enzyme initiates glycolysis in muscles, brain. It has a high affinity for glucose, so even at suboptimal glucose level in blood, the enzyme favors the glycolysis. The end product of this reaction, glucose 6-phosphate, through act as a feedback inhibitor, the overall reaction helps the expenditure of too much of cellular ATP when glucose is abundant. Whereas the counterpart of hexokinase enzyme in liver and pancreas is glucokinase, act as a higher concentration of glucose. Thus, when glucose concentration is at peak after rich carbohydrate intake, the enzyme helps liver to remove excess glucose and thereby regulate blood glucose level after meal. Moreover, the enzyme activity is not inhibited by glucose 6-phosphate. Now let's try to understand the checkpoint 2, that is phosphofructokinase. It catalyzes the phosphorylation and converts the fructose 6-phosphate to fructose 1,6-biphosphate, which is an irreversible as well as the rate-limiting reaction of the glycolytic pathway. PFK is a tetramer having 4 identical subunits. ATP is allosteric inhibitor of PFK. Hence, glycolysis is down-regulated when intracellular ATP level is high. The allosteric mechanism includes binding of ATP at a distinct site on PFK different from the catalytic site and inducing a conformational change that rotates the position of 2 amino acids, which are at position of 161 and 162. When low affinity for substrate is observed, the charges are imbalanced as phosphate on fructose 6-phosphate is repelled by glutamic acid at 161. While under high affinity for substrate, F6P results in a stabilization of charges between the phosphate at 6th position and arginine at 162nd position. This conformational change helps in maintain intracellular ionic strength and regulate blood pH, further reducing glycolysis and preventing accumulation of acid due to rigorous muscular activity under low oxygen environment. It also helps in regulating and minimizing the lactoacidosis. Other regulators of these enzymes are ATP, that is by feedback inhibition, AMP by reverse inhibition, ADP by allosteric inhibition, citrate again by feedback inhibition, and fructose 2,6-biphosphate again by feed-forward inhibition. This is a checkpoint 3, that is regulation of pyruvate kinase. This is the last enzyme catalyzed reaction of a glycolysis that is irreversible and can be regulated entire pathway. At low glucose concentration, covalent phosphorylation inhibits the pyruvate kinase activity. But if fructose 1,6-bisphosphate is formed, The reaction is activated instead, and as a feed, forward activator fructose 1,6-phosphate leads the enzyme catalyzed reaction in forward direction. Other regulators are AMP and ADP that positively regulate the reaction, while ATP is negative effector of the reaction. This slide explains the account of energy, that is how much amount of energy is liberated during the glycolysis. First of all, let's try to understand at which step the energy is consumed. Step 1, hexokinase step, 1 ATP is consumed. Step 3, phosphofructokinase step, again 1 ATP is consumed. So total 2 ATP is consumed. Now, energy is produced during the glycolysis. At step 6, 2 ATP. At step 10, at pyruvate kinase, 2 ATP. So total molecule of 4 ATP is produced and 2 ATP is consumed. So net ATP molecule is 2. So students, let us summarize what we have learned in this module. Regulation of hexokinase. Hexokinase is the enzyme which converts the glucose to glucose 6-phosphate. Hexokinase is responsible for glycolysis in muscles, in brain, and has a greater affinity for glucose, so even at lower glucose level, it favors the glycolysis. Means glucose concentration is available in limited amount, then also this enzyme favors the glycolysis. Glucose 6-phosphate is the end product and act as a feedback inhibitor of the overall reaction. Checks expanses of ATP. Means this glucose 6-phosphate, which is the end product of this reaction, is able to control the path. The case is reversed with the glucokinase, which is present in the liver. Do remember this, glucokinase is found in the liver cells, which removes the excess of glucose from the blood after heavy carbohydrate meal. So when we eat a lot of food, so blood glucose level rises. So glucokinase that is present in the liver will remove the excess of glucose from the blood. It regulates the blood sugar level. Second aspect, that is a regulation of a phosphofructokinase. Phosphofructokinase catalyzes the phosphorylation and converting the fructose 6-phosphate to fructose 1,6-biphosphate. Do remember this, glucose to glucose 6-phosphate. Now this step, that is fructose 6-phosphate to fructose 1,6-biphosphate, is catalyzed by the phosphofructokinase, where ATP is allosteric inhibitor of the phosphofructokinase. Hence, glycolysis is down regulated when intracellular ATP level is high. So it means that when we have enough energy in the form of ATP, we do not require glycolysis. So ATP actually inhibits the phosphofructokinase. Third aspect, that is regulation of pyruvate kinase. At low glucose concentration, covalent phosphorylation inhibits the pyruvate kinase activity. But if a fructose 1,6-bisphosphate is formed, the reaction is activated instead as a feed forward activator. Fructose 1,6-bisphosphate leads the enzyme catalyzed reaction in forward direction. Other regulators are AMP and ADP that positively regulates the reaction, while ATP is negative effector of the reactions. So over here what happens? Adenosine monophosphate, adenosine diphosphate concentration is high. It means that ATP is less. So they are positively regulating, means they promotes the glycolysis. While if the ATP concentration is high, we don't need to go for glycolysis, and that's why it acts as a negative effector of the reaction.